Today, we are going to talk about targeted therapy and precision medicine. In the 1970s, the US started a war on cancer. This was spearheaded by Sidney Farber and President Richard Nixon. At that time, cancer is becoming more and more prevalent in the population. And scientists from early success of cancer drugs felt that if there are enough effort and funding provided, in 20 years, we could conquer cancer. There was tremendous excitement. Unfortunately, despite the funding, the private foundations from public funding sources, cancer treatment has not been very successful. So in 2015, cancer accounted for 1 in every 6 deaths in the world. And in developed countries such as US, it's estimated that 1 in 3 in the population will develop cancer sometime in their lifetime. And so it's a very significant social and economic issue. The reason the war on cancer has not been as successful was because in the 1970s, people didn't really understand the mechanism of cancer very, very well. And so in the last 2 lectures, we talk about projects such as TCGA. Through the molecular profiling of tumors, scientists are now understanding the cancer mechanism a lot better than before. And traditionally, cancer therapies, probably the earliest approach is surgery. You just cut, right? And then there's radiation therapy, you burn. And the excitement from Sidney Farber was chemotherapy. He was one of the pioneers in chemotherapy. It's really just using poison. And it's actually poisoning both the healthy cells and the cancerous cells. And the patient get very sick, but physicians, scientists are just hoping that the chemotherapy will kill more cancer cells than they kill normal cells. So hopefully it's a race of which one get killed more. And so these are, non-specific, cytotoxic, and mostly these chemotherapies affect fast dividing cells. So normal cells get sick, but they don't die as much because they are not dividing as fast. Whereas cancer cells, because they are dividing, they need more resources. So they are potentially more susceptible to a chemotherapy poison. But that's kind of the weapons doctors or scientists have in the 1970s or 80s. What has really changed cancer treatment is the advent of targeted therapy. These are drugs that inhibit a specific target, and so they are less toxic to the normal cells. Many cancers are, in the previous 2 lectures, we see these oncogenes. They can be mutated or amplified in the cancers, and they drive the tumor growth. And in some studies, scientists call these tumors to be addicted to the cancer driver genes. And therefore, if we can inhibit the driver gene, we can kill the cancer cells and leave the normal cells intact. So today's lecture is mostly about target therapy. We will discuss immunotherapy in the next 2 lectures, and also there are new modalities such as cell therapy or RNA therapy for people who hear about the Moderna or Pfizer COVID-19 vaccines. These are RNA therapies. We won't have time to discuss these new modalities, but today we'll just discuss targeted therapy. At the end of the last lecture, we mentioned that there are different hallmarks of cancer. There could be uncontrollable death, evasion of the immune system, uncontrollable growth or replication, and immune evasion or promoting inflammation, or activating metastasis, inducing angiogenesis to provide more blood vessel and bring nutrient and to the tumor. And also because they're also genome instability, therefore the tumor can evolve very quickly and they resist cell death. And also the energy metabolism can really change. So all of these kind of hallmark work together, but in each case, there's usually a driver. And so if we understand the right drivers, we can use a drug. So in the inner ring are the mechanisms of tumor development and progression. On the outer ring are the different therapies that were developed to target the different drivers of these tumor hallmarks. So we will just mention a few examples here. The earliest targeted therapy is tamoxifen. So this is a small molecule that it can interact with estrogen receptor. So if you remember the early ChIP-seq lecture, estrogen receptor is a transcription factor. Normally it takes the estrogen hormone to interact, to bind to the estrogen receptor, for estrogen receptor to be activated. Then it can bind to the DNA and as a transcription factor, turn on nearby genes for expression. And some of the ER target genes can really drive the, in this case, breast cancer cell proliferation. So 2/3 of the breast cancers are called ER positive, meaning that their estrogen receptor either have overexpression or overactivity due to an excess supply of the estrogen hormone. And so overall, the ER become more activated. Tamoxifen is this small molecule which can block, it can also bind to estrogen receptor, so but block the ER function. So the normal estrogen hormone can no longer bind to the ER because they're being blocked by tamoxifen. And so this is a very effective treatment. And because this is approved in the 1970s, the drug is now off the patent. And so it's a very inexpensive and very effective drug treatment. Another targeted therapy we want to mention is Herceptin. So we mentioned 2/3 of the breast cancers are ER positive. They're also about 1/4 of the breast cancers, they are HER2 positive. So HER2 is a growth factor, a growth hormone receptor. So normally there needs to be external stimuli. So you can see here in normal cells, this is the cell, this is the nucleus, and on the cell surface, there are usually growth hormone receptors. They need external signals to stimulate the growth. For example, when you have growth hormone during early development, the cell needs to proliferate, they need to grow, or in very early fetus, there are a lot of growth hormones so that the cell will proliferate. So in order for these receptors to stimulate the downstream effect, there needs to be a hormone signal. But sometimes in the cells, there could be mutations which cause these factors to be activated without the receipt of external hormone signaling. And in terms of HER2, it's interesting that the hormone for this is unknown. Instead, in HER2 breast cancers, we see an overexpression of the HER2 receptor on the cell surface, and they together stimulate the cancer cell for uncontrollable growth. And so with this discovery in around the 1980s or early 90s, people started to develop an antibody drug. And so in terms of targeted therapy, there are small molecule drugs or antibody drugs. For small molecule, in this case, the small molecule can infiltrate the cell and can go interact with factors inside the cell. Whereas for HER2, there are also small molecules that bind to this little pocket and stop HER2 function, but these drugs are not as effective as another class of drugs, which are antibodies. antibodies works by binding. These are antibodies are also proteins. They work by binding to another protein and also kind of block its function. And they only work on cell surface proteins. In this case, the HER2 is a cell surface protein. And when you have a HER2 antibody, this antibody can bind very strongly to the HER2 protein, and it can block the HER2 function. And so trastuzumab is just one such HER2 antibody. They are also called Herceptin. There are later generations of antibodies, such as pertuzumab, which prevent HER2 protein from interacting with HER3, because sometimes these two can form a dimer and also activate downstream signaling. So there are antibodies that can, like bind very strongly to HER2, therefore blocking HER2 from interacting with HER3. There are also other antibodies such as cetuximab or panitumumab. So these antibody names are kind of interesting. They also, kind of block the HER2 interaction with EGFR. And there are also things like TDM1. So this is an antibody that carries a little toxin. So the star in here indicate a toxin. They are called antibody drug conjugate. So you use a little linker to bring the poison to the cell that have a overexpressed HER2 on the cell surface. And so it's a very targeted way. Basically, the antibody provides the location, but then also provides the toxin that specifically delivered the drug to the cells that have overexpressed HER2. These are really effective because the HER2 positive breast cancer sometimes have like 100-fold or 50-fold overexpression of HER2 protein compared to normal breast. Therefore, the antibody only kills the HER2 positive breast cancer cells and leave the normal cells intact. Another example is Gleevec. This is kind of a very important example also for targeted therapy. So it's a similar situation for so it's a kinase, tyrosine kinase. Normally in chronic myelogenous leukemia. And so there is a small molecule that was discovered. This drug, initially the pharmaceutical company, so initially it was developed in a small biotech company called Ciba-Geigy. Later on, it was bought by Novartis. Initially, Novartis was reluctant to develop this drug because this disease is fairly rare. Every year, there are only roughly 8,000 individuals who will develop this cancer. So it takes tremendous amount of resources to develop a drug. And if there are only a few thousand patients who can benefit from the drug, sometimes pharmaceutical companies are reluctant. But there was a reporter whose mother developed this type of disease. And so he started a campaign from like PR to force the Novartis to develop this drug. And turns out this drug works like miracles. And so, Novartis was able to get this really approved in 2001. And it's kind of an example of a drug that, the pharmaceutical company developed in a more altruistic way, but turns out to be really effective. And you can see from the clinical trials in here, traditionally with chemotherapy, the survival of the patient was only about 30%. But with the Gleevec drug, 90% of the patient can live. This is a drug that does not cure cancer. The patient has to be on this drug forever, but they can actually maintain a pretty normal life. And so the mechanism of this is for the Philadelphia chromosome, which happens in about 95% of the CML, the chronic myelogenous leukemia. So in about 95% of CML, there's a fusion between two genes called, one is BCR, the other is ABL1 or ABL. So the BCR ABL, one is from chromosome 9 and one is from chromosome 22, and now they are merged together. I think BCR provides the high expression, so it's kind of highly expressed. And ABL actually is kind of a growth driver. So normally these two are in different locations. BCR can be highly expressed, it's okay, and also ABL normally is turned off. But from this fusion now, there's a very strong expression, but also ABL now is a kind of a kinase, which can activate cell growth. And so in this fusion, because the kinase now have overactivity, it can phosphorylate the downstream substrate. And then there's kind of a cascade of signaling, which activates cancer cell growth. The Gleevec drug, this small molecule that was developed, is able to block this interaction here. So it blocks the ATP binding domain on the kinase of the ABL, which basically without ATP, the kinase cannot work to phosphorylate the downstream substrate. And so this way the tumor, the cancer cell cannot really proliferate. This discovery of the Philadelphia chromosome as a driver for CML was made 40 years before this Gleevec drug was developed. So you can see here, understanding the biology to developing a drug still takes a very, very long time. Of course, this is, back in the 1960s to the 1990s. In recent years, because of molecular biology, because of all the things that are happening on things we discuss here, today you can see that the drug development process is becoming faster and faster. The last example you can see, it's an example of things moving much faster. This is the ALK inhibitor. So ALK is also a kinase. It normally functions in the brain. Interestingly, there is also a fusion similar to the BCR-ABL. The ALK, with the fusion, ALK expression is activated and they also have more activity. And this is kind of a kinase, and downstream you can see there are many, many different pathways that are activated. The RAS, MAC pathway, the PI3 kinase, AKT pathway, just many growth pathways. And this first rearrangement or gene fusion was discovered in 2007 in Japan. And so this is a very fast discovery. And once it was discovered in lung cancer, non-small cell lung cancer, scientists were just trying to look for a drug that can specifically target this type of ALK translocated lung cancers. It turns out many of the kinases in the human genome, they share a similar kinase activity, the ATP binding domain. And so the result is that a lot of the kinase inhibitors that pharmaceutical companies develop have cross-reactivity. So there was a small molecule which was initially developed as a c-MET inhibitor. It's another kinase. And so this inhibitor initially was developed to inhibit the c-MET. It turns out it's really effective in inhibiting the translocated ALK in non-small cell lung cancer. And so it was kind of used then as a ALK inhibitor now instead of a c-MET inhibitor. And the clinical trial for the ALK inhibitor started in 2010. And because it was so effective, it was approved as this drug called crizotinib in 2011. So you can see now with molecular biology, because sequencing or nowadays you can get, 2017 is probably not from sequencing, but now you can see if we understand the molecular changes and there are fast ways to screen for drugs, we can develop a small molecule targeted therapy much, much faster. And so this is kind of examples of targeted therapy development. With the recent development of high throughput sequencing, there is a much faster from bench to the bedside translation. Um, because in the early days, researchers do their molecular biology experiment in the lab, and doctors have their separate meeting with other doctors. There was not a lot of crosstalk, but because of high throughput sequencing, patient tumors can be directly sequenced. Scientists can look at these mutations, and those help understand which patients respond to what drugs, and then that can very quickly inform the doctors on how the patient can be treated. And because of high throughput sequencing that became available in the 2007, in the last decade, we see many major US hospitals starting to do tumor profiling programs. In fact, for hospitals like Dana-Farber Cancer Institute and Memorial Sloan Kettering Cancer Center, their patients are, every cancer patient after diagnosis will get a mutation profile. And so this is not really sequencing their whole genome. At least at Dana-Farber, we sequence only a few hundred genes. These are usually driver genes that are frequently or recurrently mutated, and anything that have a known drug or have a clinical trial that's potentially targeting that mutation is in the mutation profile. And some other mutations, even without the drug, as long as it's a recurrent mutation, they are in the tumor profiling panel. And so the Farber one probably have like a few hundred mutations. Recently, Dana-Farber also started on a selected basis do RNA profiling of the tumors. And the goal is that based on the specific mutations the patient carries in their tumor, the patient can be then matched to a particular drug, which will only kill the cancer cells carrying that mutation, but spare the normal cells. And so the patient are getting this mutation profile for free. And for example, the case of non-small cell lung cancer, almost 40% of the patients can be matched with a targeted therapy. Therefore, that mutation report is very important. If the patient has a mutation with a targeted therapy, then they may not need to be treated with chemotherapy, which is really toxic, and patients very often have very bad tolerance to the drug. And so even though they are unlucky to develop cancer, if they are matched to a targeted drug, they are kind of luckier at least. And sometimes patient can be matched to a clinical trial even for a new drug that's under development because they have a mutation with a new drug targeting it. Unfortunately, still most of the mutations have no known drugs. So out of the hundreds of mutations that we profile, only minority of those mutations have a known drug that can target them. And also tumors very often develop resistance to targeted therapy. These are especially true for metastatic tumors. Very often you can have a drug, you use it for a while, and then the patient will develop resistance. So let's show this, I'll show you this example. So this is in melanoma. There are often 40 to 60% of the melanoma have a mutation in the BRAF gene. This is another kind of a mutation that causes a whole kinase cascade of signals you can see here. After, like BRAF is here, upstream there's RAS, and downstream there's ERK, or MAC-ERK, and so these are kinases. And then they activate a transcriptional program to start cell proliferation. And so 40 to 60% of the melanoma carries a mutation called V600E. So on the 600 amino acid, the V valine is mutated into a E. I don't remember what this, another amino acid. And so there is a small molecule inhibitor called vemurafenib, which can really block the enzymatic pocket at this mutation. Because of this mutation, the the BRAF can be constitutively active without an upstream growth signal, and the small molecule can just inhibit that. And this is an example of a phase 2 clinical trial. You can see here 50% of the patient or 53% of the patient with the BRAF mutation can respond. They really benefit. Unfortunately, the duration of the response is fairly short, only 6 months. And the overall survival of those patients is 15 or 16 months, which means that even though a lot of patients, this is an example of a paper that's published at Dana-Farber Cancer Institute. This is unfortunately only a 38-year-old male with the BRAF mutation and melanoma. You can see this is their melanoma. This person have so many tumors on their body. When they are treated with the BRAF inhibitor, in 15 weeks, it's really like a miracle. All the tumors disappeared. This person is like normal now. Unfortunately, 23 weeks after the therapy, the tumor has grown back, and eventually this patient passed away. And so a lot of the targeted therapies, the patient can stay on for a while, and even if they continue to use the drug, they benefit, their life get extended, but it's only for a short time. So by the way, before we move away, I want to show you something. It's notable that in this figure, A and C, if you look at where the tumor appears in C figure, even though in here you do not see any tumors left, when they do grow back, they grow back at almost the same spot. You can see there was a tumor here, and now it's here. There was a big tumor in here, and they are here, right? There are 3, 1, 2, 3 in here, and there are these 1, 2, 3 tumors here. And so it seems that the kinase inhibitor hasn't completely killed the cancer. When the tumor grows back, it's growing back at the same place. So it probably means at this point, the drug was able to kill probably 95% or even 99% of the cancer cells in this location. But the remaining 1 to 3 or 5% of the cancer cells that are resistant to the drug gradually grow back. And because they are no longer being controlled, whether they're resistant to the drug in the first place, they grow back at the same location. And so this is a table showing you the progression-free survival of many different targeted therapy. Progression-free survival means from when you see the drug having an effect to the drug still maintaining the tumor growth inhibition. How long can this drug be used before the tumor starts to grow back again? That's the progression-free survival. And so you can see, that for androgen receptor in prostate cancer, there's like one new drug which works for 5 months. In breast cancer, this works for 5.8 months. And you're like, oh, didn't people use tamoxifen for forever and it's really effective? For the non-metastatic tumors, tamoxifen is very useful, and sometimes surgery was already taking the effect, and tamoxifen is only used as an adjuvant setting to make sure there's not any residual tumors near the primary sites. Those are more effective. But for the already metastatic tumor, you can see a lot of these new drugs that takes years and years to develop, and these drugs are also very, very expensive. They are only able to extend this is also the average time of the progression-free survival for the drug. Even for HER2, for trastuzumab, you can see here, the effect is only 13 months. Although there are sometimes really lucky patients, their tumor are completely cured. But the average is not very, very exciting. And so, in fact, we mentioned here, metastatic tumor developing drug resistance accounts for about 90% of the cancer death. Patients don't die from primary cancers. They die from metastatic tumors. And once the tumors become resistant to the drug, then, if there's no additional drugs that can kill it, then the patient will die. And if we look at the different BRAF inhibitor-resistant tumors. So over the years, when the patient developed resistance to a drug, in many research institutions such as Dana-Farber, we would collect the patient tumors. In this particular case, you can still collect the patient tumors and then do either mutation profile or RNA expression profile to see what are the changes that cause this tumor to become resistant to the drug. And it turns out there are many, many different mechanisms. Sometimes you could see from the same patient multiple metastatic tumors. If you just test their multiple lesions, you would see there's multiple pathways come up to evade or resist the drug. And so far, there are a number of resistance mechanisms. So for example, this is, say, a signaling from outside. And if this is mutated, it will have so normal cell is like this, but if there's a mutation, this kind of cell surface proteins, like usually the EGFR or HER2, these type of cells, molecules that are on the cell surface, which normally takes a growth hormone signal in order to activate. But with a mutation, they can be constitutively activated, and then the downstream signaling will be on, and the cell will start proliferating. If you have a small molecule inhibitor, it can block the activity domain of this, say, EGFR. You can block its function. But there are a number of resistance mechanisms. The first is that supposedly this drug can block an enzymatic pocket. The cancer cell can develop another mutation on that enzymatic pocket, making the drug no longer fitting the pocket very well. So the drug cannot get into that pocket anymore. This way, the EGFR can now get activated again, then it can stimulate the downstream growth. Another possibility is when this first signal is on, it stimulates RAS, there could be a parallel pathway. So there is the EGFR, HER2, HER3, EGFR is the HER1 in some sense. There are different other growth hormone receptor pathway, and they can all activate the same RAS pathway downstream. And so if this is no longer a path, the cancer cell genome is very unstable. It's trying all sorts of things to stay alive and keep growing. And so if one way is blocked, the cell will try another way, use another growth, overexpress or mutate another one on the parallel pathway to activate the downstream RAS ERK pathway. The third way to evade this drug is because this drug only blocks the upstream signaling, there could be additional mutations on RAS or on RAF. And these mutations, once they happen, they don't need anything above. They can start constitutively activating everything below. And so that they bypass the upstream activation. And the final one is there's a completely different way for the cell to grow. It does not take this pathway at all. It's just a totally different pathway, which can also help the cell grow. And so all these together can make the drug become the cancer cell becoming resistant to the tumor. This is because one of the hallmark of cancer is genome instability. Also epigenetic instability. From both the DNA level, mutations can happen, and on the RNA or epigenetic level, the cell can change their transcriptional programs to depend on another pathway or another gene for the uncontrollable growth. And so the general mechanism of resistance, there is innate resistance, which means you probably didn't profile the right patient, and they never really should be treated with this drug anyways. So they don't respond in the first place. But also there is acquired resistance. So for example, initially the tumor were like this. And as it grows, additional mutations were acquired. Some of them were passenger mutation, but other ones can be driver mutations that help the initial mutation so that the cell can grow even faster. And then when you have a drug, it will probably kill most of the cells that say supposedly initially there is a driver mutation. Maybe there's a drug that kills 99% of the cancer cells, but then there's a little clone which have an additional mutation, which becomes resistant to the drug. And then this thing will gradually grow back. And that's why the relapsed tumors very often appear in the same location as the primary, the initial, not the primary tumor, the initial metastatic tumor. If it's in here, after the drug treatment, the tumor might disappear, but actually there could be 1% cell in here, and then gradually it will grow back. You will see the same tumor on the same location. Yeah, so from somatic mutation, copy number variation, gene expression change, epigenetic changes, because the cancer cell is unstable, it's trying all different ways to evade the drug. And so in order to really identify resistance mechanism, we mentioned one way is to look at patient evolution. You could look at one patient from before and after treatment, and sometimes you can follow. Initially, they responded, and then after they relapse, you can take the relapsed tumor, compare with the original tumor before treatment, and see what changes happened. Are there additional mutations? Are there difference in expression or epigenetics? We can also look at patient cohorts. In this case, if we have a group of patients that are treated with the drug, for example, in that melanoma phase 2 clinical trials, only 53% of the patient responded. And so it would be interesting to look, compare the tumor. Usually these are pre-treatment tumors. We take their tumors and do DNA or RNA sequencing. And then after the patients were treated with the drug, you look at the difference between the patients who responded versus non-responders, and you ask, what is the difference? We can also look at the pre-treatment samples versus the post-treatment samples. And this depends on, whether the responder tumor are still present. Like sometimes you can see the tumor shrink, but it's still there. That's when you can use the surgery to take out the residual tumors and do profiling. But sometimes if the tumor is responding very well, you may not even feel a tumor in there anymore. Then you can't really take the tumors. You can only take the non-responders, but not the responders anymore. We want to show one study that was published in 2017. These are MDS patients. So this is kind of like a pre-leukemia phenotype. And the patients have been treated with something called azacitidine. This is like the one of the chemical weapons that people use in World War I. It's like mustard gas. Turns out this thing is pretty toxic, but it can cure the MD, or at least treat the MDS patients. And so the authors in this study collected the MDS from the bone marrow, and they're asking, what's the difference between the responders and non-responders before treatment and what happens after treatment. And so if we look at the responders compared to the non-responders, you can do differential expression analysis and look at their pathways. They can also look at the patient survival. You can see here when the patient is a responder, they have longer survival, but if they don't respond to the drug, they die earlier. The survival part is pretty standard. But the gene expression analysis, you definitely see significant differences. This is the pathway analysis. They look at what are the enriched pathways that are upregulated in genes in the responders or upregulated genes in the non-responders. And you can see here, they are quite different pathways. And they also did gene set enrichment based on the level of differential expression between the responders and non-responders. But you can see here, we mentioned this last module, you will see a lot of things that you have already learned earlier in the semester. Once you are given the patient expression profile, I think every student in this class should be able to do these type of differential expression and pathway analysis and understand the mechanism better. So there are some challenges in analyzing clinical cohorts. The first challenge is it's ultra high dimension. Because we are doing DNA sequencing, you are essentially evaluating thousands of genes for mutations. If you do RNA sequencing, you're also, again, evaluating tens of thousands of genes for their expression. Whereas in these clinical cohorts, I think for this particular study, there were about 20 patients there. And so the patient count is so much smaller than the gene count. So multiple hypothesis testing is an issue. A lot of these clinical studies, when people look at how patient tumor evolved from initially responding to the drug to becoming resistant to the drug, relapse from the drug, is called a case study. There is no statistical power, and you are just reporting this as an observation. So this, to really identify a general mechanism could be hard. And the second is sometimes it's very difficult to collect and profile these clinical samples. Usually it's easier to collect the samples before a treatment. For example, we say, well, do you want to get your tumor profiled? We can, doctors can help the patient select the best targeted drug that can be used on the patient. Usually the patient are kind of happy to be profiled because they can potentially avoid being treated with a very toxic and non-effective drug. But after treatment, it could be difficult to collect samples, especially if the patient is responding, especially if the tumor is in the internal organ. If it's melanoma, it's on the body, like you can just scrape it off, it's easy. But if it's in the liver or kidney, then the patient do not want to have a very invasive procedure to collect the tumor biopsy. It's possible if the patient develop a resistance after the drug treatment, they might be receptive to taking another biopsy to see whether a new drug should be used. That's okay, but it's still very difficult to take. You usually cannot. It's not like a surgery. You can take a big chunk of tumors to do many different profiles. It's usually just a little core biopsy, tiny amount, and so usually not enough to do many different type of profiles. And these are also fairly expensive, although sequencing now is getting cheaper and cheaper. And the third is, it is impossible to do genetic manipulation on the clinical samples. You can't knock out the gene or test the treatment of drugs. Therefore, we need other experimental procedures or models to do this. Okay, so any questions about the second part? Because it's very difficult and expensive to collect these samples from patient, there are models that people can use to simulate a human tumor. These include cancer cell lines. Initially, this, for example, HeLa is a very well-known one from cervical cancer. So you take the cancer cell from the patient tumor and you plate them onto plate. In the early days, people didn't have good success. HeLa was one exception. It was just so growing so fast that it was able to grow in the petri dish. But over the years, people have learned better ways to treat the cell, grow it in different media, so that there are a couple thousand different cancer cell lines available now. Another is called tumor organoid. If you take the tumor from the patient and you grow them in kind of a three-dimensional, they grow into tiny little tumors, whereas in cancer cell line are usually grown as a two-dimension in the petri dish. And so in this little organoid, usually it can mimic the drug resistance better than the cancer cell line in the two-dimensional scale. And sometimes in recent years, people are also able to grow the organoid still with the immune cells or fibroblasts near it. Whereas cancer cell line, you are only dealing with the cell lines and not the overall environment of the tumor. So these are models. Another one is a patient-derived xenograft. So this is initially from the patient tumor, and you take out the tumor and you chop them up into smaller pieces, and then you implant them in mice. These are not wild-type mice. They are often nude mice, which means that they're immune compromised, and that's how the tumor can grow in the mice. And sometimes there are studies like this where after the patient has surgery, the tumors are taken out and they can be given implanted in multiple tumors, and scientists or doctors can treat the mice with different drugs and then see which tumor can shrink in the mice. Then they will know which drug to give to the patient. In terms of growing, the patient-derived xenograft is the easiest. Most patient tumors can be grown in mice. Cancer cell lines is a little harder organoids, I think probably even harder. And also there are genetically engineered mouse tumors. So for example, scientists can create mutations either using a carcinogen or specifically create a mutation on a gene, and then this tumor will evolve. And once this tumor is there, you can take out the tumor and then plant them in new mice, and they will also grow the tumors carrying this mutation. And so these type of models are much easier for scientists to test many different genes, many different drugs, do manipulations and figure out the response mechanism. And so one example, by the way, these type of mouse experiments are still very expensive. And so most of the data available are still on the cell lines. And so in order to do this cell line analysis, usually we can grow these cells in Petri dish of either 96 well plates or 384 well plates. And then each well, you can add a different drug. And these are usually done with a robotic automation. And then we can measure the phenotype, such as growth. You can also measure the gene expression of the cell after the drug treatment, which will then give us a lot of very interesting insights. And depending on this, the scale of the screen, there are two types. One is you can test, thousands or even tens of thousands of drugs on a few cell lines. You just want to see, this is to see, what the different drugs are doing. For example, the Broad Institute have a project called Connectivity Map. They measured the expression of only 1,000 genes. They have probes to just measure 1,000 genes. And after the cancer cell lines, they have many cells, like a handful of cells that are treated with tens of thousands of drugs. This can help them, and then after the drug treatment, they can see the cell growth, but they can also measure the gene expression and by the value of those 1,000 genes. And based on the 1,000 probes, they can look at all the public data and infer the remaining 20,000 or 19,000 genes, or 20,000 genes in the genome to get a rough idea of the other genes, how they change. And so using this, for example, you can calculate the differential expressions of these small molecules. They can group the drugs into different classes because there are small molecule compound libraries that can have, sometimes millions of different compounds. You first want to see roughly what they do. And so using this type of cell line, like a small cell lines, number of cell lines, if you can measure like 1,000 genes, you can get a rough idea. Okay, this is one group of drug, the cells respond in this way. There's another group of drug, the cell responds in another way. That's kind of different from the first class. And interestingly, there is one study. This is a study from the same group that developed the connectivity map or the L1000 gene panel. They look at the TCGA between tumor and normal for some cancer type. This is neuroblastoma. They look at what genes are differentially expressed, especially upregulated in the cancer. And they ask what specific small molecule can treat, when you treat the cancer cell with this drug, the downregulated genes are the upregulated gene in this cancer type. So basically you find a small molecule that can have a maximal reversal of that disease signature, meaning that if you have this type of cancer patient, if they are treated with this type of with this small molecule, those genes will be now downregulated and potentially be used as a cancer drug. And so you can see here now, using these cancer cell markers, cancer cell lines and these marker genes, you can really identify a new drug for a disease, right? Another type of screen is we use limited number of drugs, sometimes even a single drug, but you test in many cell lines. For example, if you know that your drug is inhibiting a gene such as EGFR, and you want to see what cancer cell lines can respond to this, then you can take, so there are projects like this, 3 different projects, CCLE, CTRP, and GDSC, and they have hundreds of different cell lines. And they are treated with different compounds. These are usually compounds that people are interested in developing into a drug. The target of those compounds are usually known. And after the cells are treated with the drug, we usually don't, in this case, measure gene expression change. Instead, we measure the sensitivity in terms of IC50. So by the way, the cancer cell lines, the hundreds of cancer cell lines have already been comprehensively profiled for mutations, for gene expression, and recently a subset of those cell lines have also been profiled for epigenetics and proteomics. But all of them have expression and mutations done. And so after you treat the cell, so usually this is done in not just one drug, one cell. You usually treat the cell with the same drug at multiple concentrations. And so, sorry, we don't have dots in here. So theoretically, you should have, when the drug is this concentration, this is how much this cell is growing. And when it's treated with for a concentration from low to high, this means that when the concentration of the drug increases, eventually a lot of cells will die. For example, salt is not really a cancer drug, but if you treat the well with a lot of salt, yeah, eventually those cancer cells will die. And so you will generate a curve like this. And so in this 386 well plate, some cells will, in one well, the cell will be treated with low concentration, medium, and then that next one will be medium concentration, next will be a higher concentration, and the next will be the highest concentration of the drug. And after that, we look at the overall cell growth, and then we come up with this curve. So there is a term called IC50, which is the concentration of the drug, which can have the half of the maximal inhibitory level. And so, yeah, you can see here, it's like, yeah, what is the maximum inhibition in here? What's the half of that inhibition level? This is the concentration. Another way is to look, because the number of points might be limited, sometimes there could be individual noise on one concentration. And so people also use area under the dose response curve. So basically the overall area under this curve as a measure of drug sensitivity. But in general, whether it's IC50 or area under the curve, the smaller the value means the cells are more sensitive to the drug. For example, if the curve drops very quickly, your area under the curve will be small, meaning that even at a very low concentration of the drug, a lot of the cancer cells are dying, right? And so using this approach, because these the cancer cell lines are well characterized already, for a particular drug, we can start to develop response biomarkers. For example, this is a drug, lapatinib. And if we look at lapatinib is a HER2 inhibitor. And so if you look at the IC50 of the hundreds of cancer cell lines, if we notice that if HER2 is also called ERBB2, the ERBB2 is the official gene name. You can see here, when the HER2 gene is wild type, the IC50 is very high, meaning the cancer cells are not very sensitive to the drug. But if you have a HER2 mutation, the IC50 become much lower, meaning the cancer cells are now very sensitive. They're dying with this lapatinib treatment, right? Another example is FGFR2. Again, the wild type, the cancer cell lines with wild type FGFR2 are resistant to the drug, or at least they're not being killed by the drug, but the cancer cell lines with this mutation are much more sensitive. Another one, this is MDM2. This is really targeting DNA stability. So there is a drug, Natalin 3A. You can see the wild type P53 cells are sensitive, but the P53 mutant cells are actually more resistant to the drug, because with P53 mutation, the cancer cell can be much more mutated. DNA instability, they can develop resistance. Yeah, and so on. So you can see here, based on the mutation profile, you can look at the response biomarkers to say, this drug should really be used to treat patients with certain mutations. In addition, we can look at gene expression. This is another example on the IC50. But this is the dose response curve. And they only tested on, say, in this case, limited cell lines. And what you can see is that there is a gene called AHR. And we are drawing the cell lines based on the expression level of AHR. You can see here, when the AHR gene expression is high, this cancer cell line has a faster kind of a area or smaller area under the curve. They are more sensitive to the drug. But if AHR expression is low, it's less sensitive to this MEK inhibitor. We can also use this type of approach to identify novel vulnerabilities. This is an example that was published in Nature by Ron DiPino's lab, because sometimes the cancer cell growth needs parallel pathways in here. So for example, in order for the normal tissue to metabolize glucose, it needs enzymes, either ENO1 or ENO2. Either one of them are okay. Interestingly, in glioblastoma, this is a brain cancer, no cure. Average patient survival from diagnosis is only less than a year. And interestingly, it was discovered that 5% of those tumors contain a ENO1 deletion. It's not even a driver gene, it's a passenger gene. It's next to another tumor suppressor, which is frequently deleted. Well, not frequently, but recurrently deleted in about 5% of the glioblastoma gene. So without the ENO1, the cancer cell can still grow because it still has ENO2 to help the cancer cell metabolize glucose. But interestingly, ENO2 has a small molecule inhibitor. And when you treat the cell with this with ENO2 inhibitor, the normal cells are okay in this case because they still have ENO1 to help metabolize glucose. But the cancer cells will die because it does not, the deletion is the cell does not have ENO1 anymore. And so the ENO2 inhibitor would work really, really well. And so if, from enough cancer cell lines with glioblastoma, we can identify mechanisms like this. Right? So model systems are very important. Unfortunately, there are also some pitfalls for compound screen. A lot of these initially are associations. We're seeing that this mutation is associated with a response, or this gene expression is associated with a response. There still needs to be additional tests to really validate that this is really causal and understand the mechanism. And also, the cell line lineage representation may not be sufficient. Even with 1,000 cell lines, sometimes you still don't get a good enough representation. And if you really want to see what happens in a mutation, in this case, the FGFR mutation, out of the hundreds of cell lines, there are only 2 that are mutated. I think if you only have 1, you just cannot even be confident that this is a real mechanism. And also, if this is acquired resistance that cancer cell develop later, you may or may not see it. If the cancer cell line already have that mutation, you can find it. But these cancer cell line experiments are usually done in a matter of 2-3 days. So some of the later, like developed resistance mechanism, you may or may not be able to find it. And finally, it does not consider immune response because the cancer cell line growing in Petri dish does not have immune cells nearby. So we lose some of that information. Therefore, there are also things like genetic screens. If you can get rid of a gene in a cell and see how without this gene, how it influences whether the cancer cell can still grow, or whether, or how this cancer cell respond to a particular drug. And of course, if you just get rid of one gene at a time, it's very time consuming. Can you do this in a high throughput way? Knock out every gene in the cell in different cells and do the whole thing in one big experiment. This is called a genetic screen. And we will discuss CRISPR screen later in this module. We want to see what are the ways to overcome drug resistance, right? This is a study published by Bert Vogelstein and Martin Novak. They look at the patient tumors that develop resistance against a drug, and they see that this type of sequential therapy, you treat the patient with one drug, you wait for them to develop resistance, it's almost certain to happen. So currently, for therapy, very often people do sequential therapy. If it works, let's just stay on it until the patient develops resistance. In order for this therapy to really be successful, meaning that you have a cure, a combination therapy is probably the best instead of sequential therapy. This way, in order for cancer cells to develop resistance against two different mechanisms or a combination of drugs, it's much, much harder. And so this brings us to chemotherapy. Chemotherapy has been used for a very long time. One of the earliest chemotherapy is this drug called antifolate. This was first pioneered or used in patients by Sidney Farber at the Dana-Farber Cancer Institute at Harvard. He was using this drug to treat patients with leukemia. And initially, the baby who were treated had a really remarkable response, but the relapse was very quick, and eventually the children die. So it was only a transient response. Later on, doctors identified additional chemotherapy to treat leukemia, and each of them would induce a transient response. And so in 1964 at National Cancer Institute, Dr. Fry and Freirach, two doctors, they started this clinical trial for 4 drugs at the same time, and also very high dose chemotherapy. It's called a VAMP. So VAMP are 4 different chemotherapy drugs, and they have kind of really very high dose, and the patient gets really, really sick for a short time, but later on they actually had a cure. The leukemia later on can be completely cured. And nowadays, most patients treated with chemotherapy are treated with some kind of a combination, either a VAMP or some kind of a revision of a VAMP. Different cancer types are treated with a different combination. Because this was fairly early, and at that time, the FDA rule was not so clear. 4 drugs was allowed to be tried on patient. Nowadays, for targeted therapy, you would want to try 4 drugs at the targeted therapy at the same time, but FDA's current regulation would not allow us to do that. And so you can imagine back in the 1940s, if FDA has as strict rules as now, we might never have chemotherapy, because you have to do, try 1 drug first, then try all the 2 drug combinations, then try all the 3 drug combinations, and then all 4 drug combinations. It's really, really challenging to really make them work. And so that's why people now try to see if there are sufficient evidence from model systems or from trials to really give people some ideas for combination therapy. You can try those in cell lines or tumor models as well. But for combination targeted therapy, there are also two types. The first type was a paper published in 2017. This actually deal with population heterogeneity. So among all the patients that were developing some disease or having some mutations, it could be that some of the patient will only respond to one drug and other patient will only respond to another drug. Therefore, if you were to treat these two drugs together, you will have the, in some sense, it's independent effect or additive effect. But the reality is you only have the added toxicity of both drugs without the added benefit for each patient. The added benefit is there's not additional, no synergy for the drug. And so that's one way of, basically if one drug killed the cancer cell a little bit, another drug killed the cancer cell a little bit more, and then combination of the drug will kill in terms of tumor growth will be additive effect of the two. This is kind of less exciting. I mean, people do kind of use it, but it's kind of less exciting. Probably the more relevant situation is like this. Within the same tumor, you might have heterogeneity. And recent single cell analysis, people are starting to see this from the same patient tumor. Even from mutations, you can see a subgroup of this patient cancer cells have this mutation, but then a little branch come out, they carry new mutations as well. From single cell RNA-seq, you can also see the expression difference of the same tumor. And so maybe the green cancer cells will respond to drug 2, and the orange cancer cells will respond to maybe drug 4, and there might be additional ones. And therefore, if we are able to do mutation profiling or do single cell experiment, we will be able to see the different subpopulations. Therefore, we can kind of give the same patient multiple drugs. There, you can actually see a more synergistic effect on the overall treatment response. But probably the more effective way is to detect the tumor onset and relapse early, and treat the tumor before it becomes too heterogeneous, right? If you can detect the tumor at this stage with that driver mutation, it will, maybe one drug would be good enough. And so recently there are a lot of effort trying to detect tumor early. For example, from some blood test through circulating tumor cell DNA or cell-free DNA. So this is tumor cell. This is cell-free DNA, it's just nucleotide sequencing. And based on the mutations, you can detect the early disease or also detect early the residual disease. Sometimes you can see based on the blood, how much percentage of the reads still contain this mutation. You can see that tumor is shrinking because the blood, cell-free DNA carrying that mutation is lower in the fraction. But then maybe after a few months of that drug treatment, if you monitor the cell-free DNA again, you see, this mutation is becoming more frequent. That actually tells us there is drug resistance. Then the patient can be treated with additional drugs to kind of prevent the drug resistance from developing. So this is kind of one example of non-small cell lung cancer with EGFR mutations. The patient could be first treated with a first generation EGFR inhibitor. And so this inhibitor blocked the EGFR interaction with ATPase. But then in the circulating tumor DNA or the circulating immune cells, we can see that initial treatment, the tumor actually shrank because the mutant EGFR is decreasing. But then over time, you can see it increasing, and then you can see here the mutation is different. Well, in addition to carrying the same T790 mutation, there is now a C797S mutation. And so with this mutation, the drug can no longer fit in the pocket. And so that becomes resistant. At this point, the patient can be treated with the third generation EGFR inhibitor, which really, once it's in the pocket, it actually cannot be displaced. It can work actually even better. And then, of course, in some cases, the patient can bypass different, use a different pathway. But hopefully, if there are enough new drugs developed, patient can be treated with a new drug. And so, if each drug can prolong the patient survival for a year or 2 years, hopefully a new drug will be developed. So the patient can wait to use the next generation of drugs. Unfortunately, on average, it takes 12 years and hundreds of millions or even billions of dollars to develop a drug. And out of all the drugs that were going through the initial development, only 2% had final success. And so there's still tremendous effort from biotech and pharmaceutical companies to develop new drugs, hoping that some of them will get through and the patient can really benefit from those. But in order to develop drug, we need to identify new targets as well. So genetic screens are actually able to help us find cancer-specific vulnerabilities. For example, you might find that, cancer cells with this particular mutation are super sensitive to an inhibition against this gene. That helps us identify the new drug target. But also people can use these called synthetic lethality to identify the tumor suppressor loss. So for example, if you have two genes, gene A and gene B, either one of them can allow the cell to grow well. And supposedly if one mutation actually make the cancer cell becoming, the normal cell becoming cancerous, and if we have a drug against the other one, the normal cell, the drug on the normal cell will not really kill the normal gene, but on the cancer cell, it will die because the drug already inhibited one and the other one is already mutated in the cancer, then the cancer cell will die. So this is called synthetic lethality. And there are such drugs available in the market. For example, when there is DNA damage, single-strand DNA damage, there is a gene called PARP, which goes there to repair the damage. But if you have a small molecule inhibitor for the PARP, the single strand break, if it's not repaired, eventually will lead to double strand DNA break. And this normally if it's normal cell, is okay because the cell has way to repair the double strand damage or double strand breaks. But if the tumor has a BRCA mutation, BRCA is important for this double strand break repair. And so now in the cancer cell, not only the single strand cannot be repaired, the double strand cannot be repaired because of BRCA mutation. Then the cancer cell will have accumulate a lot of mutations and DNA damage very quickly, and then they will die, right? So that's, and so basically the PARP inhibitors are being now used in cancers with a BRCA mutation already. And this is through a synthetic lethality mechanism. And so the normal cells are actually spared in this case. So in summary, we can see that in the previous two lectures, using multi-omics profiling can help understand the tumor mechanisms, potentially identify driver genes. We can use targeted therapy to kill cancer cells based on the known driver mutations or driver genes. But cancer cells are likely to develop resistance to this targeted therapy. you can look at clinical cohorts, but probably, more recently, people are using a lot of model organisms, cancer cell lines, organoid, mouse tumors, to find the response biomarkers to understand which type of cancer should respond to this type of drug or genetic perturbation, and also understand the resistance mechanism. So if there is a mutation in this cell line, but it's not responding to the drug, if you have enough cell lines with this, you can ask what is in common for those and identify the resistance mechanism. And so the compound screens can identify the correlates with drug efficacy, and genetic screens can identify real resistance mechanism. And we'll have a separate lecture on genetic screens. And so resistance to targeted therapy is extremely prevalent. And this is due to tumor heterogeneity and also the genome instability and cancer evolution. And so the effective way to treat it is early detection and also combination therapy. And we will discuss cancer immunotherapy in the next lecture. This one also is a very exciting new direction.